Hello, everyone. May I have your attention, please? My name is James Hank, and I'm the director of the Museum of Comparative Zoology, in which capacity I also chair the Harvard Museum of Natural History Advisory Board. So I'd like to welcome you all here. It's especially nice to see so many people who share my love for calamari being in the audience. Before I forget, let me ask a favor of you, please. Those of you who have cell phones, if you could turn off your ringers. Tonight, we're very pleased to present a lecture by Professor Gonzalo Giribet on the biology and evolution of mollusks. Gonzalo's lecture marks the opening of a new exhibit, also about mollusks, which features research by Gonzalo and his lab, and some amazing specimens from the collections of two of the MCZ research departments, Malacology and Invertebrate Zoology, and zoology or paleontology? Paleontology, sorry. Including several rarely seen Mollusca glass invertebrates. For those of you who are not aware, of course, the Mollusks are the German family who produced the famous glass flowers of Harvard, which we have upstairs. Well, it turns out the Mollusks also made glass animals, of which we have a collection of about 400 or so in the building, and several of them will be on display. An important part of the core mission of the HMNH is to showcase research by Harvard faculty and take advantage of our marvelous collections, so we're especially proud of this exhibit. Following the lecture, we invite members to a reception upstairs where you can get a sneak peek at the new exhibit, which opens officially on Saturday to the general public. For those of you who are not HMNH members but are interested in becoming a member, as we say, operators will be standing by who will be happy to take your membership dues, and you can go right upstairs and join this evening. Before I invite Gonzalo to the podium, I'd like to formally, I have a few introductions to make. First, before we get to MCZ personnel, I want to acknowledge the presence in force, I'm told, of the Boston Ecological Club. So you're here. All right, okay, thank you. Thank you for coming. They are no strangers to this building. You meet here regularly, monthly, or? Yes. Okay. First Tuesday of every month. And the next meeting is when?
8 o'clock, February, March,
First Tuesday in March, okay, 8 p.m. But I'd also like to thank the people and acknowledge the people who were crucial to making this exhibit possible. Of course, Gonzalo we'll hear from in a couple of minutes, but also the students, postdocs, and staff associated with his lab, the MCZ's Department of Malacology, but especially Curatorial Associate Adam Baldinger. Adam, where are you? There's Adam. From the Department of Invertebrate Paleontology, Jessica Cundiff. Where are you? Jessica there. Actually, I'm gonna call the names that we can applaud at the end. There's a lot of people here I want to thank. Mark Brinskowski, you're there, okay. You raise your hand. Alex Ziegler, it's a postdoc. There's Alex. Then Linda Ford, Director of Collections Operations, and her staff. Linda, are you? Yes, there she is. Linda arranged for the display of the glass animals, which were recently restored by Elizabeth Brill, a conservation specialist who I saw you here. Yes, Elizabeth. And then the HMNH exhibition team, Jan Sacco, Director of Exhibitions. Actually, I saw Jan not too long ago, still up there, putting the final, are you here? Final touches on the exhibit. Right, okay. Head of Graphics and 3D Design, Sylvie Laborde. Are you somewhere? Okay, she'll be coming. The paint is not yet dry. And then exhibition fabricators Tristan Roker and Colin Kelly. So now let's thank them all, please. We have really a great professional staff here. As a museum director, I sometimes have to deal with things that are not fun. Fortunately, however, such things are balanced by the opportunity to work with some very talented, interesting people, and right at the top of my list is tonight's speaker. Gonzalo Giribet is professor in the Department of Organismic and Evolutionary Biology and curator of invertebrate zoology in the Museum of Comparative Zoology. Gonzalo is a Spaniard, actually a Catalonian, they're not the same, mind you. who received undergraduate and graduate degrees in biology at the University of Barcelona. After graduate school, he moved to the USA, where he assumed a postdoctoral fellowship at the American Museum of Natural History before he joined the Harvard faculty in 2000. I don't think it's an exaggeration to claim that Gonzalo is the world's leading invertebrate systematist. Several aspects of his work are impressive, but to me the most impressive is its sheer breadth. At one end, Gonzalo does careful, detailed studies of alpha taxonomy, discovering and naming new species of spiders, mollusks, and other invertebrates based on both sophisticated molecular as well as detailed anatomical data. At the other extreme are advanced genomic analyses that explore the most fundamental phylogenetic relationships among invertebrate animals, a subject that remains very contentious and unsettled to this day. Gonzalo applies all these studies towards his basic interests in biodiversity, biogeography, and phylogeny. He's also deeply committed to both teaching, he's a very popular undergraduate instructor and graduate mentor, and to communicating his work to the general public, as we'll hear this evening. All of these interests and topics are on display in the new exhibit, which we hope will help people, and especially non-scientists, understand the process of scientific discovery, as it is conducted today with data from every possible source, not just specimens collected in the field, but genes, anatomy, developmental biology, anything that might help solve the problem at hand. Gonzalo, please.
Thank you very much, Jim. Let me turn this on. Can you hear me now? No?
Yes.
Yes? Okay. Yeah, Jim forgot one thing. You know, I'm a scientist, and my communication skills might not be as nice as the ones from the people who have put together this exhibit. As you can see by my title, a dry scientific title, The Biology and Evolution of Mollusks, or the title picked by the staff at the exhibit department, Shell Masters of the Marine Realm. Okay? And
Excuse me, could you speak up or use the microphone? I do have a mild hearing loss and I can't hear you.
yes.
Thank you.
Okay, let me see if this is a little bit better now. No? I have a microphone here that doesn't work. And it's on?
It's on.
It's on. Okay.
Closer to your mouth.
All right. So I'll try to be a little bit louder also. Is this okay now? I'm used to teaching, so normally I don't use microphones. I prefer to be louder. So we're going to cover different aspects of mollusks today, just to give you a little hint on what are these very interesting organisms that I've chosen as one of the models to study many questions in evolution, in biogeography, in ecology, in conservation. So first I want to explain to you a little bit what are they. And I'm going to start by a tree. I do trees. That's my job, to just put together these trees that then people don't understand. What they're trying to tell us are the relationships of the organisms. And one of the most fundamental questions in evolutionary biology is to try to figure out how organisms relate to each other. Why are we interested in doing that? Because then we can go and answer a lot of different questions that we might have had before. And I'm not going to bore you with this tree. I just want to say that mollusks are somewhere down here related to other groups of worm-like animals, like these ribbon worms or these annelids, for example. Okay, so we know now where they fit in the evolutionary tree, and we can study the evolution of some of their features, like their development or their larvae or their shells, and compare them to the other organisms that are related to each other. And that's what we're trying to use those trees. But it's also showing you that mollusks are only one of the 30 many animal fundamental units that we call phyla. So it's one of 30-something, 35, 36, depends on the author. But it's not just one of them. It's actually a very important one. It's the second one in terms of diversity. We have here the representation of the largest animal phyla. Obviously, the arthropods are taking a very large proportion of all the known animals, especially because of the insects. But the second largest group of all animals in terms of numbers of known species are the mollusks. And the mollusks are the kings of the oceans, or the queens, whatever you want to think of the oceans. The next group, the third one, are the vertebrates, to which we belong, and then there's a few other groups. But mollusks are really amazing. Most of the diversity remains in the oceans, and this is why we're going to talk about some of those aspects of their life in the oceans. But I told you that it's the second largest number of organisms in terms of how many species do we know. But do we really know how many species are there? And in order to answer how many species are there, we need to know what's a species, which is a very old debate in biology that I'm not going to try to get into today. I might say a little bit about how our species recognize, and I might give you an idea of why is it so hard to determine how many species are there. How do we count species? Do we count names on a database? Names in a museum? Do we do biological studies for each species we're interested in? So these are some of the aspects that I'm going to tell you about. And we can look at some numbers, and there's one that is the total number of species recorded, and depending on the author, because there's not a single database where you can compile all those things, says that there's about 1.4 million animals that have received a name and been described. Okay? Just to give you an idea, 70% of those animals are found only in 12 countries, which means that probably there's a lot that we need to know about. Now, many African countries have very little biodiversity described, even though they probably have comparable numbers to many of the other non-African or African countries listed here. Okay? So the best we can do is not go with these 1.4 million, but try to do some estimates, and the estimates are so disparate as going from 2 to 100 million of living species of animals. If you want to count the extinct diversity, there's much more. So we're really, even scientists working on these sciences, this type of biodiversity sciences, we're not very sure of how many things are there out there. And I'm going to tell you why. And I'm going to give you an example of one particular study. This is, to me, the best biodiversity survey ever done by our dear colleague, Philippe Boucher from the Paris Museum of Natural History, where he's been studying biodiversity in many places. He has invested huge resources in trying to figure out the numbers of species for different parts of the world. Probably you cannot see this very well. This little thing you see here is supposed to be New Caledonia, a little island near Australia. So you have here a map of Australia. New Caledonia is located here. It's a pretty small island relatively to other ones. And he studied a small bay in the south coast of New Caledonia. So just one little region. And what they did is go there with a team of many scientists and try to figure out what lives there. And they recorded the number of sampling hours that they did on every station. For example, they spent about 75% of the time searching in soft bottoms and about 2% of the time finding things in hard bottoms. Okay? And they had several different stations. They used all sorts of techniques to collect things, not just picking the nice shells from the beach, but many divers sucking all the sediment from different areas and then bringing that to the lab and spending years trying to document what's there. And what did they find? So they counted every individual that they found, and just to give you some numbers here, for the bivalves they found more than 45,000 individuals that actually they identified to belong to 519 species. And for the gastropods, they got numbers of 81,000 individuals, and that was about more than 2,000 species. So they counted in that place, in a little bay, they were able to record 2,738 mollusks. Okay? That number, might say, well, we've heard there's millions of animals. Is that big? It's not big. So we will compare that with other things. The other thing that we want to see is how this biodiversity is distributed. So here you have a curve, a distribution curve of the number of species from these 2,700 species and the size range that they occupy. So you have here 1 inch, about 25 millimeters, in size. So most of the mollusks that they found are smaller than 1 inch. So most of the things that you find are things like these, which is the smallest mollusk known. And these large things, they are the ones that interest many of us. There are not many species of these things. For every one of these, there's going to be thousands of smaller things. Okay? That's also interesting. Very few species of large things, many, many species of small things. Now, not all these species have names. There's another thing that they're trying to understand, are the distributions of these species, and see whether we're recovering all the possible species in that small bay or not. And we find these typical curves of distributions of biodiversity, where you have a few species here on the left of the curve that are extremely abundant, more than 1,000 individuals of one particular species. But there's a large proportion of species for which you find 1 or 2 individuals. And in fact, for about 50% of the species they collected from those 2,700, of 50% they were found only 1 to 5 specimens. And about a quarter of those were found in a single station. Okay, this one, actually 32% of them were found in a single station, which means that if you had missed that station, you will have 32% less species. How many more stations could have they studied? Okay, this is telling us that really the magnitude of the biodiversity escapes to us. And even in areas where we know very well the fauna, there's tons of species to be found, discovered. Now, again, let's go back to that number. In that study, they found a number around 2,700 species. Is that a lot of mollusks for a small bay in New Caledonia? Well, they compared to all the studies that have been done around the world for entire areas. For example, the Red Sea. And if you make a list of all the species that have been found in the Red Sea, it's only 1,700. There are 1,000 species less in the entire Red Sea than in this small bay in New Caledonia. Is that because New Caledonia is more diverse? No, I think that it's because people haven't done this type of intense studies in many other areas. And in fact, their numbers are only comparable to numbers of entire biogeographical provinces. So all the mollusks known from South Africa, going from many different regions, it's about the same, 2788. All the mollusks known, for example, for the entire Mediterranean is only 2,000. Okay, so in a little bay in New Caledonia, they have faunas that are comparable to very large. So that tells you why it's hard to know what's out there. He's also been sampling many of those waters for a long time. And he's chosen a group of mollusks that everyone who works in mollusk taxonomy hates because they're really complicated. These are the beetles of the mollusks, the turrets. And Philip studied these animals for a long time, 20 years collecting intensive sampling in the waters of New Caledonia, going from down to 3,700 meters depth. And this is only, they calculate, about 0.3% of the world's bottoms. And in that area, they have been able to recover a number of turrets that is almost not the same, but 1,700 turrets in New Caledonia. It's almost like the whole number of species they found in that bay, just for one family of gastropods. And they're estimating that maybe up to 70% of those are new species, still undescribed, still unknown to science. Okay? So trying to estimate how much unknown diversity of mollusks is there in the oceans is really hard. And only by detailed studies like this in many different places, we might end up knowing the magnitude of the unknown diversity that we have. But there's many other problems. And I want to give you another problem, which is taxonomic problem. And many of us might be responsible for these type of problems. Now, this is a species which is pretty common in certain areas. The common name is called the striped false limpet. It's not a real limpet. It's actually more closely related to the land snails that have lungs than to the typical limpets. And this limpet was described by the father of taxonomy, Linnaeus, in 1758 and gave it the name Siphonaria pectinata. Back then, they didn't have internet. I don't know if you're aware of that. And people just described the same species again. For example, d'Orbigny in 1841 described another species called Siphonaria lineolata that now we consider to be a synonym of this one. And then Stearns came in 1872 and described another one. and another one. And in fact, there's 12 names for Siphonaria pectinata that seem to be the same. This is why I'm telling you that it's very hard to tell how many species are there. Because if we count the names, and if no one had gone and revised this particular species, we will be counting 12, or 13. But if we've done the synonyms, maybe we count 1. And in fact, that species has a pretty broad distribution, a distribution that it's not very common for marine organisms that need to live in coastal areas. And it's found from southern Spain down to the Gulf of the Biafra and Guinea. And it's a pretty, this distribution is continuous land, so it will be found, but it's also found in Florida. And in fact, I was collecting there, picked up one of these things, and told my American malacologist, what species is this? And he's like, Siphonaria pectinata. I'm like, no, you must be wrong, because Siphonaria pectinata is the one I know from Spain, and it can't be the same thing. No, it's Siphonaria pectinata, and we looked, and I found out, there's 13 synonyms, and it lives abroad. And then people are saying that it has invaded the U.S. from Europe. But some people say, no, it has invaded Europe from the U.S. It's like, well. So what do? We go and study that, right? We collect the samples in the extreme points of the distribution, and we look at the genetic patterns of those things, for example. And we did that. And with Gisele Kawauchi, a postdoc in the lab, we studied the Siphonaria pectinata from Florida, the one from Cameroon, the one from Spain. And in fact, if I look at the shells, the one from Cameroon and the one from Florida look more similar than the one from Cameroon to the one in Spain. When we do the genetics, we find a signature telling us that the populations from Africa and Europe are closely related. They have very similar sequences, but they have very different sequences with the ones in Florida. There is no way that the species from Florida has come from Europe or Africa, or vice versa. These things are probably two different species. What's happening? Okay. So, if we don't study the genetics in this case, we wouldn't know also that it's not 13, it's not 1, maybe it's 2. Okay? So, is that common? It is common. It's common even in big things. And now this is a small thing of a big thing, right? I couldn't bring the big one here. This is a Tridacna. Tridacna is beautiful shells. People have been studying these things for a long time because they're beautiful, because collectors love them. And it turns out that when people study the genetics of the Tridacnas from the Red Sea, they recognize Tridacna maxima, one species that has been known to occur there for a long time. They found Tridacna squamosa, which is also known to occur there. And they found a lot of sequences from something that looked very different. A new species of Tridacna that was described only in 2008, 3 years ago, that had been totally unrecognized morphologically, but that genetically is very distinct. And then they did some morphological studies where they could actually recognize. And it turns out that it's also giving us information about the ecology of the Red Sea through time. And they can see that this species, which is this little one here, is not abundant nowadays. It's very rare. It was actually the most common species of Tridacna in the Red Sea. And the other two ones just have been expanded recently after human settlement. Any help here? No? This is off now. Okay. Can people hear me if I speak like this? Yes? Okay, let's avoid technology. Technology is bad. There you go. So there's been a big lineage that went almost extinct in the Red Sea. It's in, going out. And this is related to human habitation of that area. Okay? And we hadn't recognized. And we have many other patterns that we can show you of undiscovered diversity of mollusks in many places. Now, I'm still telling you what's a mollusk. Mollusk is many different things. Some of them that you might recognize, like that little bivalve there, or the nautilus, or even chitons if you're, mollusk savvy. But there's things that look like worms that have no shells also. And then obviously the many beautiful gastropods, including the nudibranchs that people recognize. Now, why are they so successful? It seems that there's one thing that made them successful, and it's the presence of an external shell. Most groups of mollusks have an external shell, and now we know a lot about the composition of this shell. It's formed of calcium carbonate that can precipitate in the form of different minerals. Nacre is formed by a specific type of deposition of the calcium carbonate called aragonite. Others have calcite, and different parts of the shell might have different types of mineralization. We know how it's deposited. We have now really detailed. No, maybe off, because otherwise I'm going to be going on and off.
We have nothing. There's nothing on the recording.
Okay, recording is important. So we have a lot of information now about what's happening with the formation of the shell. And I was reading a paper today preparing the lecture where there's actually different colors that get turned on and off by different genes being expressed at different parts in this larval gastropod. It's amazing. Now we can see the genes that make the shell, how they get deployed, how they do the colors on the shell. So this is a great time to study a lot of these questions. But there's no doubt that these things have been very successful because of the presence of shell. Now, do all mollusks have a shell? Not all of them do. What they do is the capacity of depositing this calcium carbonate that forms the shell. But there are primitive groups of mollusks, like these vermiform worm-like things, these solenogasters, that instead of having a shell, what they have are these spicules that are formed of calcium carbonate, the same thing as the shell. And they have the same type of glands in their epidermis that secrete these types of structures. Okay? So probably evolutionarily there was first the capacity for depositing calcium carbonate, and later on the formation of a more rigid shell that has given them this protection and success in the oceans. There's another structure that is very interesting in mollusks, and it's a special type of organ that is deposited in the mouth, near the mouth. It's called the radula, and it keeps growing. And it's just a structure that will serve for scraping the algae from the surface of the rocks, or if it's more developed, they could serve for example, tearing the flesh out of the prey. And there's species like the conus that you might know that actually have venom, and they have transformed the radula into kind of like a needle structure that can inject the venom into the prey. So I think that between the shell and this feeding structure that is very particular, and that we know that it's been in the fossil record since the Cambrian, the mollusks have achieved their diversity thanks to that. Now, how old are mollusks? So we have here, if you're used to read some of these geological scales, this is the beginning of the Cambrian, and this is one of those trees that I was mentioning that gives you the relationships of animals. What I want to show here are the stars, and the stars indicate the first occurrence of one of those groups in the fossil record. And they are groups of modern mollusks, for example, like monoplacophorans, up there in that corner, that are present from the early Cambrian. And we also find bivalves, and we also find gastropods already from the Cambrian. We need to go a little bit later to find the modern types of cephalopods, extinct cephalopods, like ammonoids originated there, and also things like belemnites. So they're all present, so ammonoids, nautiloids, and belemnoids. They're all originated also probably towards the Ordovician or late Cambrian. Back then, the oceans didn't look at all like the oceans nowadays. You can see that in the Middle Cambrian, most of the land masses that form the continents that we know nowadays were packed towards the bottom of the world, and we had a huge ocean with currents going all over the world. And there was very different sea conditions, but these animals were already there. And they have persisted until today. Now, these are modern groups of mollusks that we recognize, but there were other things. There are things that we are not sure whether they are mollusks or not. They have some features that remind us to the modern mollusks, and others that might not. For example, in the left we have Halkieria evangelista, a very interesting fossil from Greenland sediments that has some sort of shells and some sclerites that could be related to mollusks. Another one that people have now assigned to mollusks is this Wywaxia, which is being classified for a while with the annelids, with the typical worms, but it has a rasping radula very similar to the one that we find in mollusks nowadays. And other things like this Echinoplex. There's been a lot of debate in the literature whether it's a mollusk or not. If it were a mollusk, it would be a mollusk related to those worm-like things, obviously, not the ones that we know with shells. But these animals have been known for a while. Maybe they're no surprise to many of you, but there's several recent discoveries from the last 3-4 years that are really, really amazing. And one of them is this thing called Orthosanchus reburus. It's kind of like a mollusk with a head shell and long spines that is found in the Burgess shell setting. It's something very new. Or this other thing here, Odontogryphus, which is if you take a chiton, remove the shells, and you have this kind of flat-looking mollusk that was crawling on the surface. When you look inside the mouth of this thing, you see these radulas that are identical to the radulas that you find in Aplacophorans nowadays. And these things have been present since the Middle Cambrian, long time ago. Okay? Things that they've just been recognized very recently in the literature. There's another one that might be more contentious, but it's also very interesting, and I couldn't stop putting it there. And it's this Nectocaris thing, which is some soft-bodied Cambrian animal discovered in Shenzhen that has stalked large eyes and also has these cephalic long tentacles and even a funnel that is reconstructed here that people have interpreted that these things might be maybe some primitive Cambrian cephalopods, the ancestors to the nautilus and the squids and things like that. Definitely a very, very interesting animal. Clearly something that was already swimming in the Cambrian oceans. So they're not only diverse nowadays, they've been diverse through the entire fossil record. We know more fossil mollusks probably than living ones, so it's always been that successful. The next question that I wanted to address is where do they live? Where can you find mollusks? And obviously, most of them live in the oceans, and you know that this planet should not be planet Earth, should be planet water. 2/3 of the planet's surface is water. If you count the volume of habitat, it's much, much more. The average depth of the oceans is in the order, I think, of 3,000 meters, and you can count how many things can live in that environment. Now, if you're lucky to go and dive in a tropical sea in the Indo-Pacific, you will see things like that, with large mollusks thriving all over the place. They're really everywhere that you go. They might be harder to find than the corals or the sponges in some places. You need to look maybe at night when they come out hunting or look in crevices and things like that. But there's a lot of species out there. And the size of those, some of those, is pretty large. You have here one of the giant clams next to me, and that's weighed much more than I do, that's for sure. The ocean is huge. It's not only that surface of 2/3 of the water, it just has a lot of features underneath. And mollusks are fortunate that they can live attached to the bottoms, what we call benthic organisms, but there's many mollusks that can live in the water column, what we call pelagic. They live from the intertidal to the deepest parts of the ocean, like the Mariana Trench. There's mollusks, the most abundant organisms in areas like the abyssal plains are mollusks. The most abundant organisms probably in many of the deepest parts of the ocean are mollusks. And there are areas where there's not a lot of diversity perhaps, or people thought there was not a lot of diversity, but now we're finding new habitats that also have a lot of new undiscovered diversity. And one of those habitats obviously are places like hydrothermal vents or the coal hydrocarbon seeps, areas that have been discovered in the last few decades by the use of submersibles, like for example Alvin, which is run by Hui, owned by the Navy, I think by Ram Tuhuri, which has been exploring the bottom of the oceans in many places. But these explorations often go only to the bottom, and there's then all the water column that still we need to discover what lives there. You can see here between these tube worms that there's these large muscles. Okay, these are these large muscles that we'll see they have a special type of feeding that makes them very unique. But working in the bottom of the oceans is not easy. This is some of the work that I've done, for example, in the abyssal plains of the northwest Spain. And just for the idea of sending one trawl, one device, to depths of 15,000 feet, it takes several hours of operation. And you never know whether the dredge is going to fall in the proper position or not. You might take 12 hours between you send it down, you drag it in the bottom, you bring it up to find out that you have nothing. And if you have something, you have just scrape a little bit of the bottom of the ocean and get a few samples. Many times, these singletons we're talking about, one specimen of one thing, something that you will never find anymore, that no one else has seen. So it's not easy to work there. But we can begin to estimate what is the diversity there. We have other problems, obviously, like working with pressure, and you know what happens to a normal styrofoam cup if you send it down 5,000 meters. It ends up like this, something really, really small. Or if the engineers who are designing the devices you're sending down there forget to just drill a little hole in a thick iron tube, this is what happens. The pressure just collapses it, like if it's paper. It's not easy. You need to send thousands of meters of wire that might get tangled, that the weight of the wire is much more than the weight of the device you're sending, and then the device might be just in the bottom, like if it's, again, a piece of paper being dragged. So it's not easy, but you start recovering some of the things that live there. Many of these things might be new species, others might be known. We need to start trying to understand the diversity of these things, the genetics of these things. It seems that the bottom of the oceans is a very uniform environment, and that there are species that are actually distributed along the basins of many different oceans. But we need to start testing that by using the proper tools, for example. And then we get to the surface, and this is a typical intertidal of the northeast, where you find typical mollusks that sometimes can live outside for a while. And mollusks did something that many other animals didn't do. They colonized the terrestrial environments. First, they colonized the fresh waters. So they're not only in the marine realm, many mollusks actually ended up colonizing the fresh waters. But from the 8 classes of mollusks, only 2 of them made it to the fresh waters. Only the bivalves, we had at least 4 independent colonizations, and one of them is very well known. There's a group of 4 or 5 species living around Australia that just gave origin to all this diversity of freshwater mussels. Now, it didn't live only around Australia when it gave origin to all this diversity, but nowadays it's just confined to that part of the world. And then we have the gastropods also with multiple invasions of freshwaters, and it's actually not so well understood how many have there been, because it's a much more diverse group, and no one has put a very large phylogeny of this group of animals. And then one group, only one of the classes made it to the terrestrial environments. And these are, again, the gastropods with your typical snails and slugs, and again, they did it multiple times. We even have now some sea slugs, which are completely independent from these ones, that have been documented in very humid environments of the Philippines to be able to go and hunt outside the water. Really interesting animals. I was talking that they also live in the water column and even in the surface of the water in the middle of the oceans. They don't need to be attached to a substrate to live or have a substrate. This is an example of the antenna. It's a pelagic gastropod that builds these bubble raft full of air and reproduces and has the complete life cycle there And then it has some hitchhikers here, like some of these barnacles that are attaching to it. I guess that at one point, when the weight is too much, this thing is going to fall and die. But because the shell is a heavy structure. And the shell gave them protection to live in the bottom of the oceans, but it's not very good to live in the middle of the water column. And we have a group of animals who have actually conquered the water column, one group of mollusks, which are the cephalopods, with different strategies. We have the strategy of the chamber nautilus, which is able to regulate the amount of gas inside its shell, and it can do these vertical migrations during day and night. But gradually, cephalopods lost this shell. The shell is heavy, the shell takes energy to build, and things like squids, for example, only have an internal structure, transparent internal structure. If you ever buy the squid that are not clean, for example, you will know that. And that's a remnant of the shell. And the cuttlefish has a large internal cuttle bone, which again, it's a remnant of that mollusk shell. Now, cephalopods are very interesting for many respects, and one of them, they talk about the intelligence and about the large eyes. But one of the groups of cephalopods that has fascinated people the most are octopuses, which have lost their shell completely. And they have become much more flexible, much more fluid, and they can do things that many other mollusks can't. For example, they can constrain themselves and go inside a very small cavity or inside, for example, a bottle. And there is a video upstairs where you can see an octopus just going inside a bottle. And they're really smart animals, and there's documents of one octopus be living in a little tank here, just crawling outside, going to another tank to eat the food from there and return it. Or in a marine station in the south of France, they were finding that one of the light bulbs every night ended up fused. And they didn't know what was happening until they put some video camera there. They were seeing that that bulb was bothering the octopus and was squirting water to it. Very smart. The smartest octopus of all them, obviously, is the one who predicted that Spain was going to win the World Cup last year. Although I think it wasn't smart. I think it's just like red. Because if you saw every match it did, it always picked the flag with the most red. So what do mollusks do? They're hanging out in the oceans, obviously, but there are things that are very important for the global ecology, for example. Now, one of my favorite group of mollusks are bivalves. Why? I don't know. I've been working on them for a long time, and they're very interesting for many things. People tend to think that they're boring animals because they don't do much, and they do bore many times with these food that they have there, and they hang out there many times with the siphon sticking out, filtering water. And they can filter huge amounts of water. Many bivalves have the entire internal body surface that becomes a gill. They don't have a mouth. These animals have lost the radula that characterizes all the other mollusks. And instead of that, they filter water. And they keep filtering, cleaning our oceans, cleaning our rivers, until we kill them. And you can see here, for example, one scallop with this huge gill here. So huge amounts of water that go through these mollusks. And in fact, people have quantified how much can they filter. And here's a little experiment where you can see the amounts of phytoplankton in the water until they put mollusks there and how the amount of phytoplankton just decreases. So they keep filtering, and obtaining the organic matter from the water. Very interesting group of animals. Now, some of these giant mollusks, like the giant clam, cannot do that by just filtering. So the giant clams do filter. They have a very nice gill, beautiful, but they need an extra source of energy. And we all like them because they have these beautiful green and blue colors. And those are some symbionts they have the same as corals. They're algae that they have that produce a lot of the energy that they need to grow this big. So bivalves have explored many different types of feeding, from filtering water to doing symbiosis with algae, or many of these deep sea giant clams that actually have bacterial symbionts in their gills. And again, they can grow very large. You can see here this Calyptogena in the hand of a researcher to have an idea of the size. These are animals that live in very deep waters where there's not a lot of nutrients to filter, actually, and they gain their energy from this bacteria. And in the deep waters, when there's no hydrothermal vents or seeps or things like that, there's actually very few particles in the water. When you're down several thousand meters, there's very little things that get down there. And these animals wouldn't survive, these bivalves, for example, filtering. So what they do, you find a lot of the protorangs that instead of filtering, they have little tentacles that keep grabbing the particles from the sediment and eating them. Or these ones that are the only bivalves that have turned carnivorous. They have this long siphon, and when they see a little, little shrimpy thing swimming by, they will just stand the tentacle and suck it in. So they are the only group of carnivores. And they have transformed the gill that instead of for pumping water, what they have is a chamber, muscular chamber, that they will use for pumping the animals inside. They do many other things. This is a parasitic gastropod. We know the free-living ones. But this is a little gastropod of the family Eulimid, which is on top of this sea star. You can see a detail of this. And this family, actually, many of them are parasitic on sea cucumbers and things like that. And some species have actually lost the shell completely, and they live inside their host and just feeding from them. But some of the most interesting behaviors, or the best studied behaviors, are from these nudibranchs or pistobranchs and things like that. And people really love these animals, not only because of their beautiful colors, but also for what they're able to do. Here we have a Sacoglossan. And what is this green stuff they have there? These are algae. They cultivate algae, and they can get the energy from them. And then the others, they have these structures in the dorsal part that they actually will feed on animals that might contain, for example, on cnidarians, and will be able to capture the nematocysts, which are these stinging cells that you find in the cnidarians, and they can store them in these dorsal structures to protect themselves. And there are others that are able to capture the secondary metabolites from sponges, and they can also use those things for chemical defense. Or some of them that have these aposematic colorations warning their predators, don't touch me, I might be dangerous. They're beautiful animals. They can do a lot of things, and I'm just giving you some examples. But maybe not all of you are as fond of mollusks as I am, and I need to give you some arguments also why, let's say the government should continue funding research on mollusks or something. Okay? This argument never fails. They're good. People like them. Well, Jim likes calamari better, but for me, the best thing, razor clams, no doubt. Now, economics are important, right? So these are some data extracted from a book of the U.S. fisheries, or North American fisheries, and the U.S. landings for different groups of mollusks that we eat, like the abalone, or quahogs and clams in general, mussels, oysters, things like that. Don't read all the numbers. But in 2003, this was a $672 million industry. And the U.S. is not one of the, it's not one of the largest industries here, but it is in many places. Japan is huge, for example. And there's another thing about mollusks, that their fisheries in general are very sustainable. Much more so than many of the fishes that we eat. So it's something that when there's a lot of research, aquaculture works for many of these species. So I think it's important that we really know their biology and how we can use them for food sources. But there are other things. Here, let me introduce you to Arctica islandica, the ocean quahoe. This is not the quahoe that you find in shallow waters, a little bit deeper waters. This is known to be the oldest non-colonial animal. They can live up to 500 years. They're in many places, and they're important for fisheries. But they are also having used the record, both the fossil record and the record of individual shells, to study the changes in climate, to reconstruct paleo environments, and also because of this property of living for 500 years, since that they have very different levels of antioxidants, they're being used for researching aging, for example. Now, how do people do this paleo environment reconstruction? Are we pulling your leg with this thing or not? I mean, 500 years, really? Can we really know that? Well, you can study these shells and section them. And if you look at this with a microscope, you see that it has all these lines, and each one of these lines is an annual ring, the same way that you can read the age of a tree. And there are shells that have been recorded up to 500 years. Now, this is one that is being studied very well. This one was born in 1494, was collected in 1868, and people have reconstructed things like the carbon isotopes, oxygen isotopes, and things that they have used to measure the climate along this structure from 1490 to 1840. And they actually can recognize like things like the mini ice age, or down here they have a year of really bad growth for this shell, this particular one, that corresponds to very heavy volcanic activity in the area. So we can study these things. We can date things. We can use these very old models also for things that are relevant to us. There's many other aspects of mollusks that we can study that are important. This is the giant African snail. You have a size there in the hand of one person for you to know the size of this thing. And mollusks having both studied as pests or for control for pests. In the US, that we have a huge diversity of freshwater mussels. They're very good bioindicators about the health of the aquatic environments. So they have a lot of aspects of their ecology that can tell us information about what they do. So I want to conclude the last 10 minutes talking a little bit about the research that is going on and some of the things that you might see upstairs in the exhibit. And this is the molluscan research in the Museum of Comparative Zoology. We have a very large malacological collection with more than 500,000 lots of specimens. And people ask, what's a lot? A lot is a group of individuals collecting in the same place. And it's a long history. It started in 1860 with the purchase of one large collection of terrestrial and freshwater mollusks from North America. And then we've had many illustrious malacologists working here. And some of the official curators of the collection started by Bill Clench, and then followed by Ken Boss and Ruth Turner, and now Jim McCarthy is the acting curator of the collection. It's a really important collection. It's produced hundreds of papers in malacology. Lots of students have graduated from the MCC using the material from the collection. There's publications that have been appearing for many decades. So what I'm going to show you is a little bit what I do, which is just the type of the iceberg of everything that has been going on in the institution for a long time. But talking about what I do or my associates do, it's easier for me. This is some work that Alex Ziegler is working on. He's trying to find new ways of imaging mollusks, to find new anatomical information, see how the shells articulate with each other. And he's using medical techniques like MRI to do these entire reconstructions of the mollusk. So you take the MRI, it's the same thing that they did with my knee a couple of weeks ago. But then he does the nice 3D models where you can reorient this thing from any angle, look at the internal structures, external structures. And he's using this with museum specimens that are not being damaged or dissected or anything like that. He also uses other techniques like micro-CT, microcomputer tomography. And this is a beautiful picture of the surface of a chiton, but also you can go and take a section and you can see these dorsoventral muscles or these structure inside that you can see here very clearly, which is that radula of the chitons. You have here a more detailed structure. Now he's done this without dissecting the animal, without disturbing the animal for anything. And he can put it back into the collection. It might look a little bit different color, but that's it. Because he stains them with iodine or So that's one of the things that he's doing, and there's a beautiful video upstairs in the exhibit showing some of the models and articulating them. The question I've been interested in always is the relationships of these things. We know they have a huge diversity. We've been studying the morphology for a long time, but every researcher has proposed a completely different relationship for mollusks, and that's why I'm trying to figure out what's going on there. And one of the key animals is this thing in the center that it's called a monoplacophorans. Now, monoplacophorans, I mentioned them earlier in the talk. I said that they were present from the Cambrian, from the base of the Cambrian. And they were known from the Cambrian and thought to be extinct for more than 400 million years. Until the Galathea expedition, the Danish Galathea expedition that circumnavigated the globe collecting deep sea animals, they dug out one thing that remained, undisclosed for a while because they really didn't know what it was, until Lemche in 1957 published it as a new living specimen of these things that had been thought to be extinct for 400 million years. Monoplacophorans are rare, they're deep sea, and they're difficult to collect. So a few years ago, I was lucky enough to get, I always wanted this animal, always, since I was like this. And a few years ago, a researcher gave me a piece of an animal that had been collected at 3,000 meters depth in Antarctica. 3,000 meter depth, by the time it gets to the surface, several hours, they had to sort it out. I got a piece of tissue that didn't look very healthy, to tell you the truth. And we tried to sequence DNA. We got the first DNA from any monoplacophoran, and we published a paper, but the DNA, there were contaminations, it wasn't sufficient really to do what we want. We postulated that it came a sister group to these things called chitons that have 8 dorsal plates. But the data weren't completely robust, so what we decided to do is we need to get fresh monoplacophoran, and we need to study more than just a few genes. We need to study the entire genomes of these things. So with a couple of colleagues, Casey Down at Brown University and Nerida Wilson, who's now at the Australian Museum, she was a Scripps, we set out to look for monoplacophoran. We went with the Gordon Sproul to the only place where these things are found in shallow waters, 12,000 feet. Okay? These things are, at least they're not 5,000 meters. And that's between San Diego and Los Angeles in the Cortez Bank. And we went there, and we got lucky. We set up the whole microscopes there to sort. These things are 1 to 2 millimeters, so we bring the rocks, and we need to put the rocks under the microscopes, hope that the bow doesn't rock too much. And there we go with the 10 grabs. So we sent this dredge down 10 times in the 3 days that we had, and all but 2 gave us monoplacophoran. And even in one of them, we got 13 specimens. So we got a total of 41 specimens of these things. The first time I've seen this thing alive, this was amazing. And we got a lot of information that we wanted to study their morphology. We got some oocytes there, and beautiful thing. And what we did with these animals is using some of the newest techniques for sequencing, we're able to sequence hundreds of millions of base pairs, put them all together, and compare them to other genomes of other mollusks. And there's been different hypotheses of how these things go with each other. And, the month that we were about to get this paper published in Nature, somebody else came with another paper in Nature doing more or less the same thing we did, except that they didn't have the monoplacophoran. So still our big idea there of asking what is the position of this really charismatic group of mollusks? Are they basal to all the other mollusks with shells? Or are they related to these chitons, as we proposed only 3 years earlier? Or are really related to some of these cephalopods that some paleontologists had proposed in the 1970s? Or finally, some of the older hypotheses that this is the stock that gives origin to all the other diversity of mollusks. So I'll conclude with one slide showing where we found monoplacophoran to go. This is our new tree. The names you don't see apparently here are in green, and those are all the new genomic data we generated. And the interesting thing here is that we found that the monoplacophorans were related to these cephalopods. And in fact, the monoplacophorans from the Cambrian, they had the shells were chambered like the shells of the chambered nautilus, for example. And that's why paleontologists had proposed that hypothesis. I hope you are fond of mollusks. You know, I just want to thank you for coming to listen to this talk. And especially I want to thank Jan Sacco and Adam Baldinger again for making so much fun putting this exhibit together. It's been really, really great. And obviously the rest of the HM&H exhibit department and all my lab members who've been doing all this research I've been talking about. So if you have some questions, I'll be happy to answer.